In this lecture, we will discuss about the meiotic crossing over. The meiotic crossing over is also called as germinal crossing over because it takes place into the germ cell. It is of most frequent in occurrence and it has a great genetic significance. As we know that when cell enter into the cell cycle, it has the two phases, interphase and the M phase. Okay, interphase is further classified into the three stages, that is G1 phase, that is growth phase, S phase and G2 phase. So S phase is important because here genetic material get duplicate. Now when cells enter into the meiosis, it has a two stages, that is meiosis 1 and the meiosis 2, while meiosis 1 is further classified into the prophase, metaphase, anaphase and the telophase. Prophase 1 has a 5 stages: leptotin 1, zygotin 1, pachytin 1, diplotin 1 and dikinesis. In the leptotin stage, the chromosome is double-stranded, though the strands are not visible due to the presence of nucleoprotein complex in between the chromatid. So these are the three stages of meiotic crossing over: synapsis, crossing over and tetrad stage. So synapsis takes place in the zygotin 1 of the prophase. Here you can see that this is a homologous chromosome pair which is replicated in the S phase. These two homologous pair of chromosomes are held together by the synaptonemal complex. Synapsis means the pairing of the homologous chromosome, and here you can see that crossing over will occur. Although most of the DNA is replicated into the S phase, but some amount of DNA or the fraction of DNA, that is 0.3% DNA, is replicated in the zygotin stage. That DNA is called as zygotin DNA. Now next stage is the crossing over, which takes place in the pachytin 1. Here the chromosome is in a tetrad stage, and that tetrad stage chromosome undergoes the recombination in the crossing over by the help of the enzyme. There is a breakage of chromatid segment and the exchange of non-sister chromatid segments. Later, there is a fusion in the nucleus. Last is a tetrad stage, which is happened in the diplotin stage. Okay, as we know that synaptonemal complex is formed during the synapsis, it begin to dissolve during the phase of tetrad stage, and the chromosome separates and chromatid becomes distinct at most of the point. As a bivalent chromosome appears to have a 4 chromatid, now is called as a tetrad stage. It occurs in the diplotin stage of the prophase 1. The synaptonemal attachment points between the homologous chromosome are called as a chiasmata. In latter stage, the chiasmata tend to shift to the side. The phenomena is called as terminalization. Many of them disappear prior to the metaphase 1. So this is all about the meiotic crossing over.